Multicellular animals have traditionally been divided into about 36 distinct phyla based on their shared anatomy and embryology. However, this traditional phylogeny is currently being reevaluated using molecular data. Now, animal phylogeny takes into account not just anatomy and embryology, but also morphology, molecular information, life history, and other types of relevant data. Like with other phylogenies, this is a hypothesis and it changes with new information. The text presents the 20 most specious animal phyla in Table 33.1. Most of these species phyla we'll be examining as we progress through the animals, including phylum Arthropoda, Mollusca, Chordata, Platyhelminthes, Nematoda, Annelida, Cnidaria, Echinodermata, and Porifera. Starting at the bottom, we'll first talk about phylum Porifera. This is a monophyletic group of animals that share a common ancestor with other animals. These are the simplest animals, so simple they used to not be classified with animals at all. Known as the sponges, they are in the group Parazoa, which translates to near animals. They have no symmetry for the most part, no tissues or organs. All other animals are classified as Eumetazoa. These are all the animals that have symmetry, tissues, etc. Most sponges are marine, but some are freshwater. They occur at all depths within the ocean and are the most abundant animals in the deep ocean. Most lack symmetry, though there are a few that are radially symmetric. As simple as they are, sponges are still animals and exhibit true multicellularity. They have specialized cells. Larval sponges are free-swimming, but adults remain attached, aka they're sessile or sedentary. The vase-shaped body of a sponge is composed of spicules, spongin, and cells. Spicules are needles of either calcium carbonate, usually, or silica, and spongin, which is reinforcing tough protein. Together, the spicules and spongin compose a type of skeleton, a strengthening structure for the sponge. Sponges have cells called choanocytes, flagellated collar cells that contribute to water circulation, bringing food and oxygen into the sponge and wastes out of the sponge. These choanocytes engulf and digest food particles from the passing water. Large sponges are very complexly folded in order to increase surface area, therefore increasing the choanocytes that are capable of engulfing food. Because the sponge relies on choanocytes to engulf particles of food, they are restricted to food that's smaller than one cell. So basically, we're talking bacteria. Sponges reproduce sexually via sperm being released into the water and captured by choanocytes of a neighboring sponge, then transported to the egg, or asexually via fragmentation. Here we can see that vase-shaped body. Sponges have pores called ostia, which are lined with these choanocytes that bring water into the ostia and then through the main cavity within the sponge, the osculum. As water flows through the osculum, these choanocytes can engulf tiny little bits of food that are in that water column. Phylum Cnidaria. These are the first organisms that we'll examine that are in Eumetazoa. So these are all animals that are that have distinct tissues and symmetry. Cnidarians, about 10,000 species here, are mostly marine, about 99% marine, though there are a few freshwater species. They are all carnivorous. They are radially symmetric and diploblastic, meaning that they consist of an endoderm and an ectoderm. They have distinct tissues but no organs. So they have the simplest bodies of all eumetazoans. However, they are capable of reproduction, exchanging gases, hunting and digesting prey, and distributing nutrients to the cells. So while they're still quite primitive, they are much more complex than the sponges. Cnidarians do not have a concentrated nervous system, but they do have nerve cells around the body and receptors for touch, gravity, and light. A characteristic of cnidarians are nematocysts, which are stinging cells, which, along with tentacles, are used by cnidarians to capture prey. Cnidarians have a body plan that has a single opening that leads to a gastrovascular cavity. The gastrovascular cavity is an all-purpose cavity. It works for digestion, gas exchange, waste excretion, and gamete formation. The body is made up of two layers, epidermis and gastrodermis, which comes from the endodermis. Between these two layers of tissue is the mesoglia, an acellular layer that holds these two tissue layers together. Epidermis is what you see on the outside of a jellyfish. Gastrodermis covers the gastrovascular cavity. This figure shows a cnidarian called a hydra. We can see the two tissue layers here, gastrodermis on the inside, epidermis on the outside. There are sensory receptors within these tissue layers, and we can also see the nematocysts that are used for prey capture. The gastrovascular space also serves as a hydrostatic skeleton. It provides a rigid structure against which the muscles of a cnidarian can operate. Basically, muscles can contract against it. It also gives the animal shape. Many polyp species also build an exoskeleton of either chitin or calcium carbonate for added support and protection. The major evolutionary innovation here is extracellular digestion. Food can be brought into the gastrovascular cavity and digested, so animals are no longer restricted to food that's smaller than the size of a cell. Here we can see the two basic cnidarian body forms, polyp and medusa. Polyps are cylindrical with tentacles pointing up. These are sessile for the most part. If they're solitary, they are attached to a substrate, but there are colonial polyps which move around together. The other basic body form is the medusa. This is an umbrella-shaped body with tentacles pointing down. These are motile. They are able to swim around and be free-living. Many cnidarians alternate between polyp and medusa. Some only exist as polyps, some only exist as medusa. In species that have both polyp and medusa in their life cycle, the medusa is responsible for forming gametes, sperm cells and egg cells. The following organisms are considered cnidarians: sea anemones, corals, box jellies, hydroids, manowar, jellyfish, and star jellies. And we'll look at some of these in a little more detail here. At the top of the slide, here, these organisms here, these are anemones. The bottom here, we have corals. These both belong to class Anthozoa. Anthozoa stands for flower-like. You might remember phylum Anthophyta, the flowering plants. So these are flower-like organisms. They're sessile, so they're attached to the substrate, mostly polyp with no medusa stage, which is their defining characteristics. They're quite complex with a divided gastrovascular cavity. While anemones are mostly solitary, coral are colonial. They're also characterized by having a calcium carbonate shell, and they form a nursery and refuge for many fish and protect the shoreline. We talked about coral a few chapters ago because they're symbiotic with dinoflagellates, a type of protist. Climate change and sea temperature change, sea acidification has caused these corals to bleach in many places, meaning that they expel that dinoflagellate symbiote, and they unfortunately die, which you can see here at the bottom of the page. Class Cubozoa are the box jellies, seen right here. These guys are medusa. They are very similar to a jellyfish, except they're composed of a box-shaped bell with tentacles that hang off of each corner. These are quite poisonous with long tentacles. They're only found off the coast of Australia. Class Hydrozoa, down here, hydroids, Portuguese man-o-war are good examples here. These consist of organisms that have a polyp and medusa stage. They're mainly colonial, like the Portuguese man-o-war, which is a colony of feeding and reproductive polyps that's carried along by a medusa. Class Scyphozoa, over here towards the right of the slide, these are jellyfish, and they exist mostly as a medusa. This is their conspicuous stage of the life cycle, but both stages are present. Finally, star jellies, class Staurozoa. Here a medusa is attached to a substrate by a stalk. These were formerly classified under class Scyphozoa with the jellyfish, but they are different enough to be in their own category now. Phylum Ctenophora consists of the comb jellies, sea walnuts, and sea gooseberries. This is a small group of marine organisms. They are typically very small, around 5 centimeters. They are radially symmetric, but unlike the cnidarians that they resemble, they are triploblastic, 3 layers of germ tissues. Comb jellies have 8 rows of comb-like plates of fused cilia that beat in a coordinated fashion. They are the largest animals to use cilia for locomotion. Because of these cilia, many are bioluminescent. They have 2 retractable tentacles covered with coloblasts, which are able to discharge a strong adhesive that's used to capture prey. Unlike cnidarians, they do not have nematocysts, and they are actually more complex than cnidarians with that addition of a mesoderm. However, they are often mistaken for cnidarians because they have radial symmetry and they have a gelatinous body. So we can close out this chapter by watching some lovely comb jellies swimming.
My name is Beatrice Dixon. I'm the founder of the Honey Pot. To have a retailer like Target see you and believe in you is everything. The reason why it's so important for Honey Pot to do well is so the next black girl, she could have a better opportunity. That means a lot to me.
Be warned. This is what happens when you eat too many Skittles, or sprinkles, or maybe adult brownies. Or, even better, all 3 at once. This is the ocean's majestic comb jelly. You'd think that those rippling bands of colors would be a case of bioluminescence, which so many sea creatures deploy. But in reality, this has more to do with the critter's locomotion. Like jellyfish,